I'm a veteran from the Air Force, and I used that GI Bill to start my college career. First started as a pre-vet, and then decided I didn't like to cut up animals, and I had a very good teacher who was in molecular biology who got me interested in that. Thought about ways that we could actually make plants so that you don't have to use pesticides on them, that they can defend themselves. So that interested me a lot. And so that's the direction I went. I went to Utah State University, and there I met Dr. Neil Van Alphen, where I first was introduced to the American chestnut tree. Before that time, I really didn't know a whole lot about chestnuts other than the song "Chestnut Roasting on the Open Fire." But in that lab, I learned about hypovirulence, where you have a virus that attenuates the fungus so that the tree can survive. And during that time, it was great because I learned a lot about the mechanisms of how the fungus attacks a tree. And that will play out later in my career quite significantly. From there, I went and did a postdoc down at the University of Florida, worked with Dr. Corby Kistler on a different fungus called Fusarium oxysporum. But then I was very fortunate to get a job up here at the SUNY College of Environmental Science and Forestry, where I could then return back to working on chestnut. When I was up here, I met Dr. Charles Maynard, where we started collaborating, and the collaboration really got started from a visit from some people from the newly formed New York chapter of the American Chestnut Foundation, Herb Darling, Stan, and Arlene Worsig. They came to our campus and met with Chuck and myself and asked us, is there a way we can use some of the more modern techniques to make a blight-resistant tree? And you know, the National was doing a very good back-cross breeding program, but we wanted to kind of incorporate some of the newer techniques. And Chuck and I said, sure, and that's where we actually got started. And of course, it's been a long haul. My expertise is more on the molecular side, the pathology side, trying to find the gene that will confer blight resistance. Chuck's side was more the forestry tissue culture side, and trying to figure out how do we propagate these trees, how do we get a gene into these trees. And it was a very good collaboration. This is a project that literally hundreds of people, well over hundreds of people, have contributed to. So this is not just me. This is really a team effort by many, many different people. And one slide I like to always present in my talks is this slide of why is GE, or genetic engineering, useful for restoration? And I don't put it on here to say it's the best method. I don't put it on here to put other methods down, because the other methods are great too. I just want people to understand what genetic engineering has to offer that's kind of unique and very useful for restoration. And I'll just start off by saying that you know, the trees that we want to restore need to be adapted to the forest that they are coming from, okay? And one trait that I like to point out is the trait of tree height, or, you know, American chestnut is a canopy tree. The closest one to that would be the European chestnut, also a canopy tree. Both of those are susceptible to chestnut blight. The other chestnut trees, the other species of chestnut trees, tend to be much shorter, only about, you know, a half to 2/3 the height of a typical American chestnut. So, you know, one of the ways you can make a resistant chestnut tree is to breed with these resistant varieties, but you're not only going to move over the genes for the resistance, but you're also moving all the other genes over to adapt the trees to their own location. So what I need you to know is that chestnut has a lot of genes. It has around 30,000 gene pairs. That means half from one parent, half from the other. And if you were to make a hybrid of these, such as the Dunson chestnut that you can buy at Walmart, about half those genes in there would be for from the American, half from the Asian species. And so you have this competing network of genes. You have some groups of genes that are saying, make me tall, and some genes that are saying, oh, keep me short, you know? And you mix them together, and what you get is something usually in between. And that's okay. It's okay if you're doing an ornamental tree, if you're doing an agricultural tree, something like that, that is fine. But for a restoration tree where you have to reach the canopy, where you have to compete with the other trees in the forest, that's not okay. And that's the reason why the American Chestnut Foundation actually instituted a back cross breeding program to try to regain those traits that make it more adapted to the forest, okay? But genetic engineering has a unique way of doing this. And I just want to use my book example. Let's say the genes in the tree are like the words in a book. And this book describes the American chestnut. Again, if you did a hybrid, half those would be describing an Asian species and its adaptability to where it came from in the world. The other half would be for the American. So you get somewhere in between. But if you're going to do genetic engineering, what happens is you have a book and all the words are the original, okay? The original for the American chestnut. And I'll just pull out a passage from a book. It says in this passage, it's very exciting in that season to roam the then boundless chestnut woods of Lincoln. So everything's in here is about chestnut. And what we do in genetic engineering is we just add a couple words to that. So now everything is still the same, reading about American chestnut, and then it goes, is very exciting that season to roam the then boundless blight-tolerant chestnut woods of Lincoln. So what we're doing is making a very small change. We're not adding in all those other genes that we then have to get rid of, and therefore the tree should be totally adapted to a forest. So you get 100% American chestnut with blight tolerance. So that's the big advantage with genetic engineering. Now there's going to be some disadvantages, which I'll tell you just in a minute, that we have to go back to breeding to deal with. But what gene do you put in there? Well, there's a lot of options. The one that seemed to work the best for us is a gene that encodes an enzyme, oxalate oxidase. It comes from wheat, but it's actually a ubiquitous enzyme. If you look at the three sisters, the main crops that grow well together from the Haudenosaunee Indians, you got squash, corn, and beans. Two of these actually have oxalate oxidases in them. Okay, so if you look at some of the published literature and you look at where oxalate oxidases can be found, you find it in all kinds of cultivated foods, crops. So you eat this all the time. It's also found in a lot of wild plants, as well as fungi, mosses, and even some bacteria. So this gene isn't anything new to the environment. So that's the gene that we're actually putting in. And what it does, it basically detoxifies an acid that the fungus uses to attack the tree. And we know this is actually from hypervirulence studies, where they found that hypervirulent isolates of the fungus can't produce this acid as well as the virulent form. So that kind of gives the first clue that this acid was important. But here's a way you can actually get rid of that acid without having to use a virus to do so. So you just give the tree a gene that can make this enzyme. This is not a pesticide, and that's very important. It does not kill the fungus. So if you're not killing the fungus, you're not killing off and only letting the ones grow that can overcome this type of resistance. So it's going to be a very stable type of resistance. So even the ones that can't overcome this still survive and replicate and maintain into population. So that's selective pressure to overcome the oxalate oxidase gene, and now they can coexist. Now, how much resistance is actually there? Well, it's actually very similar to the Chinese. So on left, there is a stem from the Chinese chestnut that's been infected with the blight fungus. On the right is a wild type American infected with a blight fungus. And you see the big difference there. On the left, you got callus, you got the infections being stopped. On the right, you have sunken canker, the tree dies. In the middle is the actual one with the enzyme being produced, the oxalate oxidase. And you see it's very similar to the Chinese with just minimal damage. So there's not no damage, but there's this minimal damage and the tree survives. Okay, so one of the disadvantages with genetic engineering is that you start off with a clone, okay? All the trees are genetically identical. And so you have to use breeding to get the diversity back into the population that now contains your blight tolerance gene. So we do this by outcrossing, and sometime I think Jared's going to be on here and he can tell you a lot more about this. You might have heard it already. But basically you take the pollen from the transgenic tree, you outcross to wild type trees, you collect their offspring. Half of them will contain the OXO gene and be blight tolerant. We have an easy assay for that. and you just keep going, and you have different complements of the original both parents, and you keep going out crossing until you finally get a very diverse population. Okay, so we're going to be big suppliers of pollen. We've already gone through 3 generations in collaboration with the American Chestnut Foundation. This whole idea of half the offspring not containing the oxalate oxidase gene is very important for conservation. And because these trees live for 100 years, 200 years, whatever, let's say 100 years from now, you have a tree like this one here. You can take that tree, you can outcross it, and if this was one of our original transgenic trees, half the offspring could be non-transgenic. So in 100 years, 200 years from now, we might have much better techniques. So we can actually go back to the original tree and modify that, as opposed to having to modify the one that we've already modified. So that's very good for conservation. So I'm just going to end my portion here with this philosophy. This is actually a quote from Robin Kimmerer from our college when she spoke at the UN. It says, "We humans are more than consumers. We have gifts of our own to give to the earth." And that's the way I view this Chestnut project. We as humans, we've actually caused this problem with the blight, because we brought the blight over from halfway around the world. But so I think it's really up to us to solve that problem. And that's been my philosophy, kind of the giving back of these trees. Okay, so